off, right? Now we want you to turn your cell phones on. However, there's a very important thing when you turn your cell phone on. We want you to turn your ringer off. Okay? So if you have a cell phone and you can turn the ringer off but just keep the buzzer on or whatever, that would be great. If you do not have a cell phone and you've got kids in the kids ministry over there, you can see there's a whiteboard set up over there. And if you can kind of keep your eye, peeled here and there, I know you want to look at the talks and stuff, but if you see someone travel over there and write a big number on there, and that coincides with your child, then of course you need to go and take care of that and see what's up there. And even, it's hard to keep looking over, but every now and then just look over, and if you see the number that has to do with your child, then please run over just so the people in the kids ministry can get things handled. Okay. I think we're here. Rob, obviously, this is a talk that needs to be delivered because I've had so much problems getting you here. So I'm not going to say anything else. Dr. Rob Carter.
Thank you. For those of you who don't know me or those who might be watching on video, I'm Dr. Robert Carter. I started this project that I'm about to describe to you with the ultimate goal of seeing how much of Adam and Eve's original genomes could be reconstructed with the current data that we've been collecting over the last decade. And what you're about to hear is a summary of the path that I've taken toward that goal. I haven't finished. I haven't even started to do half of what I'm planning, and there's probably more to do that I haven't even realized yet. But I just thought that what I've already learned might be a tremendous help to many people. Now, this is a big deal. In fact, a person came up to me just a minute ago and said, "Hey, did you hear the news report from today?" I said, "Which news report?" He says, a professor at Trinity Western University, which I think is in the States, just was on record. He says Adam and Eve never existed. There's absolutely no way. Well, Francis Collins has been saying the same thing. If you don't know Francis Collins, he was a director of the Human Genome Project in the States. President Obama appointed him to be the director of the National Institutes of Health. So this man has a very high-profile Christian, claims to be an evangelical, which I don't doubt. But he's also the founder of BioLogos, which is an organization specifically dedicated to promoting theistic evolution, as in evolution is absolutely true and creationism is not. Now, Collins has been making a lot of press lately. In fact, one of the reasons for my opening here is because over the last several weeks, we've gotten multiple emails, questions after church talks, questions at homeschool events, questions at seminary talks, just questions all over the place. People keep asking us this question, but this guy over here says that Adam and Eve didn't exist. Well, and a series of articles appeared in Christianity Today in June. Christianity Today, a very widely read magazine, said this. It says, Collins' 2006 bestseller, The Language of God, A Scientist Presents Evidence for Belief, reported scientific indications that anatomically modern humans emerged from primate ancestors perhaps 100,000 years ago, long before the Genesis time frame, and originated with a population that numbered something like 10,000, not 2 individuals. Christianity Today. Same article. In a recent pro-evolution book from InterVarsity Press, put those two thoughts together, InterVarsity Press and pro-evolution, that's very interesting. The Language of Science and Faith, Collins and co-author Carl W. Giberson escalate matters, announcing that unfortunately the concepts of Adam and Eve as a literal first couple and the ancestor of all humans simply do not fit the evidence. Now, he's also been saying this in the verbal word, not just in print, recently at Pepperdine University. There is no way you can develop this level of variation between us from 1 or 2 ancestors. And then NPR, National Public Radio in the U.S., just recently, in fact, just last week, I think, yes, last week sometime, they had a series of interviews with people involved in this issue, and Francis Collins says similar things, but so did this man, Dennis Venema, who's one of the fellows at BioLogos. He said, you would have to postulate that there's been this absolutely astronomical mutation rate that has produced all these new variants in an incredibly short period of time. Those types of mutation rates are just not possible. It would mutate us out of existence. Are you feeling intimidated yet? Good. I meant to make you feel intimidated. However, not everyone is buying into this line. Al Mohler, the president of Southern Baptist Theological Seminary, venerable institution in the States, very important place, he said this. He said, the moment you say, in the same NPR article, by the way, he said, the moment you say we have to abandon this theology in order to have the respect of the world, you end up with neither biblical orthodoxy nor the respect of the world. Richard Dawkins agrees with this. He said on Revelation TV back in February, he said, I think the evangelical Christian, now this is a Christian radio station he's being interviewed on, and he says this is a very interesting thing. He says, I think the evangelical Christians have really sort of got it right in a way, in seeing evolution as the enemy. Whereas more, what shall we say, sophisticated theologians are quite happy to live with evolution. I think they're deluded. And he laughs. I think evangelicals have got it right in that there's really a deep incompatibility between evolution and Christianity. Now, that statement in itself doesn't mean that theistic evolution is wrong, but it does indicate that if you're seeking to curry favor with the world, theistic evolution doesn't work. Now, here's the root of the problem. I strongly disagree with Dr. Collins's statements. Now, I have to do this carefully because he is a Christian, claims to be a brother in Christ. I must treat him differently than I would treat a strict atheist. However, I do believe that he's got his cart before his horse, and he's using evolutionary assumptions to draw his conclusions. Because the statements that he have made come straight out of evolutionary mathematics. It doesn't appear that he's ever considered what the Bible would predict and what the results would be from a biblical prediction. He's specifically appealing to the assumptions of deep time, common ancestry, and the idea that mutation plus drift plus selection is the only thing that you should use to explain human diversity. Now, granted, he's basing his statements on a lot of data, a tremendous number of data, including the Human Genome Project, the ENCODE Project, which is a program where they did a massive sequencing of many active regions in the genome, the HapMap Project, where they sequenced millions of single letters from thousands of people, and I'm going to spend a lot of time talking about that in a minute, and then the ongoing 1,000 Genomes Project, which they have done a number of genomes in this project so far, but the goal is to sequence 1,000 people's genomes worldwide, so we have a comparison of all these different things. Now, let's talk about HapMap. Now, okay, is everyone okay with what I've said so far? I didn't insult anybody, did I? Just is what has been said out in the thing. I'm not attacking this person. In fact, I'm just going to say this at the end, but I'll say this now. I understand the position of Francis Collins. I was there. Many of you were there. All of us have a very difficult time fitting science and faith together. And it's a lifelong process. We have to work on it. You don't go, poof, of course, I know all the answers. And the world is saying things, and it takes us time to work through things. So I'm going to focus now. I'm going to focus on one particular set of data. It's called HapMap. It's a massive database. I actually have it right here on my laptop, so it's not that massive. But the goal of the HapMap project, excuse me, I just ate some nuts. I got some nuts stuck in me. The goal of the HapMap project was to identify and catalog genetic similarities and differences in human beings. Now that excited me tremendously because I said, that's the kind of data that I need to look for Adam and Eve. Because the Bible's true, making the right predictions about Adam and Eve, we should see the data today. Well, what they did is they took over 1 million letters. There's an estimate, there's about 10 million very common variations amongst people worldwide. About 10 million. Well, they looked at over 1 million of them. They selected a group of them. So these million letters, these million places in the genome are variable. So at position number 52, some person has an A, some other people have a G. At position number 1,000,003, this person has a C and this person has a T. At position 2,052,000,052, there's an A and a C. So they looked at variable places in the genome, one letter at a time. And they sequenced these million letters, over 1 million letters, in more than 1,300 people from 11 world populations. Very exciting to me because now we're talking Tower of Babel. However, the word on the street is that this does not support the biblical data. Now, granted, at first pass here, I ran some analyses. Here, I took the HapMap data, and I just counted up the number of different types of variations. How many places in the HapMap data are there A's and G's? How many places are there C's and T's? How many places are there A's and C's? How many places are G's and T's? And this is the summary. So that's 1 million data points up there. Now, looking at this, at first blush, I said, oh, that looks like something I might expect over millions of years of evolution. Watch. Now, I'm going to give you two things that I might expect over a million years of evolution. I'm not going to contradict my statement, so don't fear. These are called transitions. A and G and C and T. This is in DNA. I put up a picture, but I didn't think about it. There are two types of nucleotides, purines and pyrimidines. One of them has a single ring, and the other one has a double ring. And it's much easier chemically to switch between purine and purine, or pyrimidine and pyrimidine, than purine to pyrimidine. These are purine to pyrimidine swaps. This is 3/4 of the data. This is 1/4 of the data. That's actually what we'd expect, because if you look at mutations that are occurring in living organisms today, transitions are much more likely than transversions. Transitions, transversions. And so this looks like a genome that has suffered millions of years of mutation, because these are more common. At random, this is what I would expect. In fact, why are these two exactly balanced? Because if you have a change in one strand from an A to a G, the other strand has to change from a T to a C. And because mutations should be random, it happens on this strand, and this strand, or this strand, over millions of years you get an evenly balanced picture. That's true, actually, for these also. These two is not true for, but I believe because the letters C and G are highly conserved in the genome, it's much more difficult to mutate those particular letters. Okay? Point number 2, that's what I just said there. Point number 2, you can take this data and analyze it in all sorts of different ways. So here I'm looking at only the people of European descent. It's called the CEU population, actually people living in Utah. Because the Mormons are very interested in genealogy and genetics, they have a lot of gene centers in Utah. And so this is people living in Utah of European descent. And what I did is I looked at chromosome number 22. That's the smallest chromosome. And I looked at only places where some people have an A and some people have a G. And so in genetic data, G is green, A is black. And what I did was, I said, what percent of this population has a G and what percent has an A? And then I sorted them all. So that you can see that there are some genes or some alleles where most people have a G and a few people have an A. There are some other places where most people have an A and maybe 1% or less of the people have a G, and everything in between. So there's some places where 40% of people have an A and 60 have a G, or 60 have an A and 40 have a G. Is that okay so far? Okay. Now looking at that, that's nearly a straight line. I did that for another population. This is the Yoruba people that live in West Africa. The Yoruba is a large tribe in West Africa, and they sequenced over 100 people in this study. And it looks very similar to the last one, but actually it's interesting, there's a bias. There's more G here than A. This line has been pushed down. And I looked at that. I stared at this chart that I generated myself for 3 months, saying, I'm dead. Because this, to me, looked like millions of years of evolution. Because over time, you'd expect new mutations. By definition, a new mutation is going to be very rare. It's only in one person, right? Well, to get mutations that have changed in frequency, either through selection or just random drift to some higher level, it would take millions of years. And so it looked to me like, oh man, look at this, and look at this. This is evidence of millions of years. And it took me 3 months before I said, oh, wait a minute. Maybe this isn't true. What would it be like if we had started with Adam? Could we get that curve? And then I had this talk all ready to go. And John Sanford and I were sitting down Sunday afternoon. We're having a brainstorming session. And he had his laptop open, and I had my laptop open. And we had these two charts up, two different charts. And it made no sense. And all of a sudden, a collective brainstorm hit us, a lightning flash. And we're like, oh, look at that. That's not evolution. That's Adam. In my coral reefs talk, you heard me quote the Duke of Argyll, who wrote an essay in 1887. He's talking about Darwin's theory of coral reef formation, how he thinks has been discredited. And he says, even though this theory has been around for 40 years or more, he said, and here we learn the important lesson that an hypothesis may adequately account for actual facts, and yet nevertheless, may not be true. Now, here's why I don't think this is true. I'm going to introduce a term to you. I've used it a little bit already. It's called genetic drift. Genetic drift is the random changes in gene frequency from one generation to the next. And I'm going to illustrate it with this chart. I did this on Microsoft Excel, and I use a random number generator. And what I did is I have a population of 20 people, which is ridiculously small, but drift happens very quickly in really small populations, so you can see it very, very soon. And in this case, let's say it works for mitochondrial DNA, which are inherited from the mother. It works for Y chromosomes. It also works for last names, if the woman takes on the husband's last name. But let's just say this is Y chromosomes. Here I have 10 people, 10 men. Each one of them has a different color, denoting each of the Y chromosome lineages. And in my small population, I said each one of these families can have 0, 1, or 2 boys. Ignore the females, don't worry about the girls. They have 0, 1, or 2 boys. Well, if they have 0 boys, that Y chromosome lineage is not passed on into the next generation, correct? It just disappears. There's no boys. If they have 1 boy, you get a copy. But if you have 2 boys, you doubled the frequency of that. And so you can see that in the first generation here, we're missing a couple. The next generation moves a couple more. And on and on. And look at this. We're losing things. And by the time we get up to here, there's only 2 left. And through random fluctuations, at generation 12, 50% of the population is of this blue type, a light blue type, and 50% of the population is of this dark blue type. Okay so far? At this point in time, in a small population, something called fixation is about to happen. All of the other lineages are going to be lost, and one lineage is going to be stuck or fixed. Fixation is almost guaranteed in small populations, and over evolutionary time, even a moderately sized population, over millions of years, random genetic drift should fix alleles, even in an evolutionary population. But at this point in time, each of these alleles, each of these different Y chromosome types, is equally represented in the population. Each one of these has a 50% probability of becoming fixed and a 50% probability of becoming lost. So in a population, the probability that an allele will be kept is exactly equal to its frequency. Okay? Now, I'm not trying to be some genetics professor just overwhelming you with stuff. I want to walk you through this process here. Now, what John and I were looking at were the results of a program called Mendel's Accountant, which is, to my knowledge, this is the most sophisticated evolutionary modeling program ever written. Of course, it was written by creationists because we wanted to, not me, but because they wanted to model evolutionary theory with real numbers. Now, if you type in Mendel's Accountant on Google, you'll get a whole bunch of blog entries, a bunch of people that hate this program, say it's stupid and dumb, but in reading their comments, it's obviously they have no idea what they're talking about. And that makes me happy. But if you go to mendelsaccountant.info, you can download this program and run it on your PC. What you're looking at right here is the typical results of a typical model run using a human-like population over long periods of time. What these are mutations. These are mutations that have occurred in the population since the run started. Now, forget whatever might have been there in the beginning. These are just new mutations. You can see that most of the mutations are rare because they have to appear in a single person. But not only that, most of these rare mutations are going to drift out of existence. Most of them are lost. Most new mutations are lost. It takes a long time and a lot of work for random drift to get mutations into some high frequency, even as much as 20% of the population have an allele. This took several thousand generations to get here. And this is a slowly growing population. It works for static size populations. It works in many different parameter settings. We see this pattern. Many extremely rare variations, very few variations up here, because if these variations here are less than 50%, then they have a high likelihood of being lost. It's impossible to stay on this point. If a variation got up here to 50%, it's going to be pushed this way or this way. It's like there's a hill here. And the variations are slipping through drift this way, and if it gets to more than 50%, then it slips through drift this way, and eventually we might get some that reach fixation. This is not the HapMap data, is it? This is a straight line where there are many variations at the 40, 50, and 60% level. We would not expect that given evolutionary numbers based on that. That was, to me, a eureka moment. I was trying to give the evolutionists a benefit of the doubt when I saw the HapMap stuff. Okay, look, Mr. Evolutionist, granted, it looks like millions of years, but it also looks like Adam, so both of us are predicting the same thing, but I no longer do that because it does not look like millions of years. It looks like Adam, and I'll show you why. This is Adam. I wrote a program in Perl. I wanted to model human populations. And so I said, okay, I can't work with a billion variations or even a million. I'm just going to use 1,000. That's a pretty good estimate. And so what I did was I gave Adam 1,000 variations. But because Adam has 2 of each chromosome, if one chromosome has an A, the other one has to have a G. Can you see that if I treat Adam as a population, all of my alleles have to be 50-50? Now, I don't know if Eve was a clone of Adam or not, but for now, let's just assume that she has all the same variations, just to make things simple, and later on we can consider the alternative, but we won't need to. Let's just copy Eve and throw her in here also, and now still, all the variations in the population are 50-50. Now, we all learned in high school biology that if you take 2 heterozygous individuals and you breed them, then this Punnett square, you're going to get 25% of the children are going to be AA, 50% will be AG, and 25% will be GG. That's a 1 to 2 to 1 ratio. You all remember that? So therefore, every single one of Adam and Eve's children would look like this. Or at random, assuming no linkage for the moment. I know genes are inherited in chunks, but I'm just making this simple. Assuming no linkage, 25% of every single child is going to be GG, 25% AG, and 50% of all those alleles will be AG. Each child is radically different than Adam. Right? Radically different. They have lost fully half of the diversity in Adam is not represented in this child. But every child, these genes are going to be inherited at random, so something that might be GG in one child might be AA in another child. So if you take this and you pool it, this is the results of 2 children combined. This is the result of 4 children combined. By the way, for you math nerds, I'm following Pascal's triangle. 1 to 2 to 1 is the 3rd level. 1 to 4 to 6 to 4 to 1 is the 5th level. It's the number of children times 2 plus 1. But 4 children, 16 children. What are you seeing? You're seeing that the more children Adam and Eve have, the better sample, forgot about that thing, the better sample we have of the initial allele diversity in the population. If Adam and Eve could have 1 million children, this curve would be a perfectly flat line. You'd have an exact representation of one generation to the next. But they can't have 1 million children. They're limited by biology and time. And maybe desire. So no matter what, and that when you go from Adam and Eve to the next generation even, I haven't even talked about the generations after that, you get a tilting of the allele frequency spectrum. Can you see that? That's not the HapMap data, is it? It's not a straight line, is it? But it's one step in the right direction. Now I thought to myself, okay, well, what if we took some non-variant place? And here I've padded, this is the same curve, but I've added 100 alleles here and here that are invariant in Adam. And what I'm doing is I'm allowing for places for mutation to occur. And as mutations occur, if this A turns to this G turns to an A or this A turns to a G, you're going to see the curving like this. And look, this is what I'm trying to get to. I'm not quite there yet. But you see what's happening. A genetic drift plus mutation over time, and all of a sudden I'm starting to look like HapMap, but not quite. So, sorry, here's what I'm supposed to explain my program. So I needed something more powerful. So I wrote a program in Perl. And I said, let's start with Adam with 1,000 heterozygous alleles. That means 2-letter variation. Now let's assume for now Eve is a clone. She has the same variations. And now, as they start having children, I'm going to assign the children spouses at random from the population. So boys and girls, let's say, I tell them when they're born, I say if they're a boy and a girl, and I let them mature. And then once they get to the age of maturity, I say, this boy, go find a random girl. And I pair them up. And I say, mating for life. And interestingly, most population genetics program, they don't have overlapping generations. They use discrete generations because it makes the calculation simpler and takes up less memory. But I have overlapping generations, meaning that a boy could marry his great-great-great-great-great-grandniece, his sister, or his great-great-great-great-aunt. Not his grandmother, because that would be a direct line. That woman would already be married. But I think it's purely biblically reasonable. Adam and Eve are going to be popping out children. Well, their children are going to get married. They're going to start popping out children. And you're going to have lots of overlapping generations if you're living for that long. Now, I don't know the basic life history statistics for the pre-flood population. I don't know it at all. But I have three main parameters in my program. One is the age of sexual maturity, when I allow a person to get married. Another one I call reproductive senescence. This is when the female goes through menopause and she stops having children. And the third one is how far apart the children are spaced. Now, each one of those actually turns out to be not very important, but the combination of those three tells me how many children per female, and that controls genetic drift because that controls how fast the population grows. So here I'm going to run a model. And here's my three numbers. I'm assuming people mature at age 50. That a female goes through menopause at age 350, and there's 20 years of spacing between each child. Now, that sounds kind of ridiculous, doesn't it? But I do that because I can't have more children than that per generation, because at 800 years, my computer runs out of memory with 100,000 people. You can do amazing things with computers if you have the horsepower. Well, HP laptop, I mean, it's a pretty modern laptop, but it couldn't handle more than 100,000 people. Fair enough. So here's my results. Remember before I had Adam and Eve, black and green? Well, here I'm replacing that black and green because I have to do multiples of these with just a straight red line. Adam and Eve, 0.5 every single allele. The population 100 years later looks like this. Not too surprising. 100 years after that, the population looks like this. 100 years after that, the population looks like this. 100 years after that, 100 years after that, 100 years after that. Where's this going to stop? There. Look what happened. In my exponentially growing population, genetic drift stopped once the population got above a couple of hundred people. Because once it got above a couple of hundred people, that next generation is going to be a little bit larger. Well, they're grabbing almost all of the genetic variation, the previous generation, and popping it into the next generation. That means the current world population today, how many people are in the world? 6 billion. Genetic drift isn't operating. Or if it is, it's so slow, and even in geological time, it's not going to occur. I mean, mathematically, it's possible, but it's going to be really slow. But this is only part of the biblical model. I'm only slightly there. And I say, wait a minute, I can start with Adam and Eve and get this, but I'm not always as far as I want to be. So, well, what's next in the biblical model? It's the model of the flood. What happens at the flood? 3 full sibling brothers and their wives. I don't know how closely related the wives are, so I'm going to pull 3 random females out of the population, giving evolution the benefit of the doubt. 3 full sibling males, 3 random females from the population, they form a new population. That's called inbreeding. What does inbreeding do? It leads to genetic drift. So here's my model again. Same exact parameters as before. Same exact parameters. There's Adam and Eve. But this time, 100 years later, 500 years after Adam and Eve, I'm going to throw in a bottleneck. Now, I didn't wait till 800 years. I wanted to do 1,500 years, but I couldn't. My computer couldn't handle it. So I did it at 500 years before my computer locked up. And I erased all the data and I could start building a population again. So at 500 years, I'm reasonably confident genetic drift is not going to occur anymore. I think that's an okay assumption. Boom, genetic bottleneck. 500 years after the genetic bottleneck, let's do it again. Boom. 500 years after that, it looks like this. Let's do it one more time. Boom, what are you seeing? You're seeing the HapMap data. Now, I'm assuming exponential population growth, which might be true, but I'm also assuming random mating, which is ridiculous. Random mating doesn't happen in any world population, or we would all be middle brown color with brown eyes. And we're not. Humans do not randomly mate, and we have not randomly mated throughout all of human history. Period. But also, is it fair to model multiple population bottlenecks when the Bible only talks about one, Noah's flood? Actually, yes, it is. Because it doesn't matter what happens to the world population. It only matters what happens to Noah's family. It's Noah's ancestry that counts. Nobody else counts. And if you think about people in the pre-flood world, they're marrying, they're having children, they're having children, they're having children. If those people spread out, they're going to be lost to one another. There's not going to be any random mating. If there's any, and there's lots of violence in the world. Well, if we have any kind of violence, we might get clannishness. We might get family snobbery. Might get geographic separation. Kings forming and kings demanding that they marry only some people and excluding others. Whatever. If there's any degree of non-random mating in Noah's lineage, this occurs to some degree. Now, the reason I'm throwing out multiples is because I don't know to what degree, but I do now know that it is possible, and that excited me tremendously. But look at something else very interesting. See this down here and up here? These are alleles that have gone to fixity. Some of Adam's genetic diversity might have been lost even prior to the flood. I had never considered that before. Some of the genetic data, the information that God put into Adam and Eve, could have disappeared at the flood. So Adam and Eve might have been even more genetically robust than we are today. Okay, something else you can do with the HapMap data. Here I am, I'm comparing the allele frequency. I looked at the entire genome of the Europeans and the Yorubans, and I looked at only the A to G variations again. There's other ones also, right? There's 1, 2, 3, 4, there's 6 classes, but I just look at A to G variations. And Microsoft Excel can only generate 32,000 data points, and it crashes when you try. So I selected 10,000 out of all of them at random so I could graph it. Even that was extremely slow. I'm very frustrated. I could do better graphs in the 1980s with Ami Pro than I can with Excel in the 2000s, but okay, forget it. This is the frequency. Each one of these dots is an allele. This is the frequency of the allele in the European population versus the Yoruban African population. What do you see? You see a bunch of mess, right? But look real carefully. Do you see that there's a giant cluster here and here? In fact, 95% of the data is within those 2 lines. 95% of the data is here. Oh, there's a trend. Yes, there's a trend. Why is there a trend? Why is it that the frequency of an allele in one population is a pretty good predictor of the frequency of the allele in the other population? Because they came from the same source population. These people, these 2 different dissimilar people groups, and you can do this for any one of the pairs of the 11 world populations, they came from a single source population. Now, the evolutionist says, this is the out of Africa event, which actually is an ad hoc explanation that was added after the fact because it was not a direct prediction of evolutionary theory. However, this is a direct prediction of creation theory. If this wasn't true, we'd be in trouble. These people came from a single common source population. Now, genetic drift has occurred. About this much, actually, has occurred between the allele frequencies have changed in one population versus the other population because at the Tower of Babel, we divided the population up into clans, and each clan got a subset of the genetic diversity. It looks like most of it, but only a subset. And then since all that time has passed, more changes have occurred. Okay? Your head's full yet? Now, HapMap. What does that mean? No one's asked me yet. Map, everybody knows a map is, right? Hap stands for haplotype. Let me explain what a haplotype is by first showing you a genotype. This is a genotype. It's a string of nucleotides. It's a piece of DNA. This person's genotype is ATG, TAG, CCC, TAG, CTG, and etc. There's 3 billion of those. Most of that information, though, is repetitive, is actually useless because most all of us are invariant across most of our genome. So why sequence all the invariant stuff that everyone has the same? Let's just look for the variable places in the genome. And HapMap, they sequence over 1 million letters, and on average, the letters are 2,800 nucleotides apart. So here I'm going to pack them in a little closer. And if I was to look at the HapMap for this person, their haplotype, not their genotype, but their haplotype would be TGATT. Another closely related person, their haplotype might be ACATT, meaning there's an A in this position and a C in that position. So not only did they skip a lot of genetic data, which is not much data because there's no variation between people, but they only took some of the variation. They ignored some of it because they couldn't do it all. But even so, there's over 1 million letters, and it saturates the genome. You can do a lot of statistical tests and tests of population studies based on these 1 million and more data points. It's really very exciting. I have some notes here. I want to look at my notes to make sure I said everything. These notes I don't think I'm going to say. I'm going to say that later. Okay. Now, the exciting part about the HapMap data is this. It was designed to be able to test for crossing over. See, when you have 2 of every chromosome in your cells, during meiosis, during the sexual reproduction process, your chromosomes, which you inherited from mom and dad, they line up, and crossing over occurs, and then you get a mixture, and you pass on a mixture of mom and dad to your child. So they inherit a mixture of grandma and grandpa along each chromosome arm. Now, it's estimated that for the long chromosome arms, there are some short arms, but for each one of the long arms, there's about 1 crossing over, perhaps as many as 2, per generation. There have only been 150 generations in all of human history, perhaps as many as 200 generations. That means that there are not 1 million of these blocks in the human genome. Oh no. In fact, if the human race was millions of years old, you'd expect it to be scrambled to oblivion. Because crossing over is supposed to be random, and it's supposed to be so many generations, but they're not. There are places in the human genome that have never crossed over in all of human history. You have Adam or Eve or Adam plus Eve on your 2 chromosomes. Not only that, but we are now discovering what controls crossing over. Not only is it not random, but it's controlled by genetic factors, and these genetic factors vary from one person to another. Well, if they vary from one racial group to another racial group or one subpopulation to another subpopulation, that's going to mean that crossing over might occur at different rates in different populations. That right there might explain why Africans have so much genetic diversity compared to the rest of the world. Perhaps they have more crossing over. But see, that's what I was trying to get at, and I haven't even gotten there yet because I got into all this other cool stuff. Oh, that's next. Maybe someone will help me on this. I don't know. Okay, let's talk briefly about mutation. I think I'm going to say this on Thursday also. Where does mutation come from? What is a mutation, first of all? It's a spelling error in your genomic encyclopedia. Very good. You heard my talk yesterday. Okay. Well, interestingly, this is from a paper by James Crow in the year 2000. He's asking the question, where do mutations come from? And since he reasons that there are between 1 and 3 mutations every time a cell divides into 2 cells, that mutations must come from cell division. But interestingly, most mutations don't come from the female, according to this paper. They actually come from the male. But you see, after an egg is fertilized, it undergoes several divisions. And after approximately 30 cell divisions, a female baby's ovary is finished. The eggs are in place, and they remain in stasis, in a suspended state, and harbored and nurtured for sometimes 45 years before ovulation. It's not true for the men, though. See, after about the same number of cell divisions, our little cells stay in stasis until puberty. And then they start dividing like crazy, and they divide until we die. And if cell division is a recipe for mutation, the older the male is when they have a child, the more mutations they might pass on to their children. This is Crow's data. So a 15-year-old man, might give 30 or so mutations, but a 50-year-old man can give about 800 mutations to his children. Wow. What happened if you live to biblical ages? Noah, 10,000 mutations. If some very old man, many thousands of mutations. this is actually kind of scary. Now I want to show you this chart. You've probably seen pictures like this before. This is the lifespans of the patriarchs before the flood. Well, I'm going to change it here. This is the lifespans of the patriarchs just compared to each other, not on the timeline. Of course, Enoch walked with the Lord and was no more. This is you see this curve, this drop off, which a lot of Christians have talked about before. This is how old the men were when they had the next one in line. So Adam was this old when he had Seth. Seth was this old when he had Enosh. Boom, there's Enoch, and there's Noah. You see how odd Noah is? Now, I don't know the age of puberty of these men, but let's say the age of puberty was somewhere around here. That means that these guys have hardly gone through any cell divisions, and Noah has gone through a whole lot. In fact, let's get rid of the ages entirely and just look at it this way. Here's how old they were when they had their child. Can you see that Noah might have been genetic poison to the future of humanity? There were more cell divisions in Noah, because he was over 500 years old when he had Shem, Ham, and Japheth. The Bible says that. There are more cell divisions within Noah than occurred in all of human history prior to him. So we talk about decline of lifespans of the patriarchal age. Well, maybe it was just from mutation accumulation. Maybe it was from some loss of genetic diversity in Adam, etc. Now here is using modern data, assuming a 15-year-old puberty and the same rate of cell division. If I apply that to the biblical patriarchs, each of these points is one of those biblical men, there's Noah. Now, if puberty sets in later, then Noah actually becomes a greater fraction of the whole. He had possibly a lot of mutations to the population. That's right, nothing appeared here. And so what I did here is this is the same data. This is the same data. And here I just put some modern averages, and I graphed it every 10 generations or something like that. So this is just modern numbers applied to the biblical thing. I know it's kind of a chimeric thing. But I don't know how many mutations people carry today. Somewhere within that cone is what I kind of expect. So people today, from Adam, were picked up between 80,000 and maybe 150, 200,000 mutations from Adam. That's what all of you carry. And I expect it's going to be different in different lines. I don't expect it to be the same everywhere. Now, if there are approximately 10 million common variations within the human genome, interestingly, people carry a significant proportion of that total. They carry about 4 million each. In fact, I'm about to say that actually, aren't I? Okay, what I did actually is I looked at the HapMap data. And I counted up the number of variant places. I looked at the homozygous alleles and heterozygous alleles. I counted up the heterozygous alleles in each person. And each person had about 30% of the total. In fact, population to population was almost the same in all populations. Each person, each population even, had about 30% of all the possible variations was in every single person. Well, if that's out of about 1 million. If there are about 10 million common variants, that means each person carries 3 to 4 million, all of you, 3 to 4 million heterozygous alleles. Now, Craig Venter, he competed with the Human Genome Project and the private company. He also had a genome sequence. And later on, he had his genome sequence, personally, both copies of each chromosome, and they counted up, and there are 3 to 4 million variations within Craig Venter. So it's 2 different independent tests of the same number. Very interesting. Now, on top of this 10 million, there are many singletons, private mutations that only occur in one person, or rare mutations that only occur in one subpopulation. There are many of those. But interestingly, the HapMap data, by focusing on alleles that are found in all world populations, they actually focused on created diversity accidentally. That's the next slide. Somewhere, I'm going to say that somewhere in here. All right. If there are 150 to 200 generations since Adam, we can actually estimate how many mutations people carry. This estimate is a pretty good number, 60 to more than 100, but 100 seems to be the general trend of multiple evolutionary papers. There are about 100 mutations per generation. In other words, you were born with 100 mutations that your parents didn't have. And they were born with 100 mutations that their parents didn't have. Your children were born with 100 or more mutations that your parents didn't have. Now, a new paper came out in June or July when they actually looked at genomic data and they counted up mutations that came from the mother or the father. And it was very interesting that Crow's data looks a little bit out of place now, but this paper said about 60 mutations per generation. And some families, most all the mutations came from the man, and some families, most all the mutations came from the woman. As maybe people have defective repair enzymes. Don't know. But 150 generations times 100 mutations per generation is about 15,000 mutations per individual. That's less than 1/100th of 1% of all the variation in the human genome. Therefore, if this is true, most variation in the human genome is that variation that God put into Adam. And all of us since then have accumulated a small fraction of mutations. This is one reason why we can carry so much heterozygosity. The evolutionist looks at that as mutation. They say all genetic diversity is due to mutation. Well, wait a minute. What if God engineered stuff in there? See, if God put any kind of variation within Adam, that variation should have been in the pre-flood population. It should have been on board the ark. It should have been at the Tower of Babel, and it should be spread about amongst us all today. And it looks like at least half and possibly most of the variation within people follows that exact pattern. Only a fraction of it should be due to mutation. Adam and Eve absolutely, actually, and just as one data set, absolutely fit the evidence. The current distribution, that HapMap data, that the 45-degree slope, is what we would expect given drift alone. We don't even have to appeal to mutation. We don't have to appeal to natural selection. The HapMap data is what we'd expect just given Adam plus genetic drift and a small population with the flood and the Tower of Babel. There it is. Not only that, but all the common variants found in the world today could fit within one starting couple. I'm estimating that maybe Adam had about 10 million variable locations. Well, you right now have 3 to 4. Big deal. Obviously, you're normal people. Even despite the fact you have all those bad mutations, the evolution, you're just fine. Adam would have been just fine too. This is not an intolerable load, especially if God created the variation. It's not mutation. It's perfectly acceptable alternate readings that work. We don't have to invoke mutation as the sole source of diversity. We don't have to appeal to deep time, common ancestry, or mutation, drift, and selection to explain this data. And I'm sorry, but that is one of the most exciting things that I've ever learned. Now, Darwinism has been called the universal acid, dissolving everything. And if we look at society, this is true. But evolution actually is the single best false explanation of world history ever invented by man. It is not something to scoff at or treat lightly. It is a powerful explanatory force, which is why so many people believe in it. And if you start with evolution and ignore a couple of assumptions in there, man, it goes a long way of apparently explaining the world. It's not something we can just blithely discard. That took a lot of work, by the way, to get through all that. That took years of work, and it wasn't all me that did it. But evolution is also far superior to Zeus standing up on top of a mountaintop throwing lightning bolts down to randomly mess up people's lives, right? So we need to approach this soberly and deliberately, realizing that we are just as liable to make mistakes as the evolutionist. And we are just as liable to allow our biases and preconceptions and ignorance drive our conclusion as the evolutionists, because we're all people. And most of us, I'm sorry, are pretty dumb. And I tell you what, considering some of the things that I was foolishly assuming, I'm really thinking that I am not so smart. However, we should take heart, because there is a universal acid even more universal than the universal acid of evolution. Look at Hebrews 4:12. It says, the word of God is living and active, sharper than any two-edged sword, piercing to the division of soul and of spirit, of joints and of marrow, and discerning the thoughts and intentions of the heart. That's the word of God. It can break the evolutionary spell, and I know that from personal experience. 2 Corinthians 10, verses 3 through 5 says, for though we walk in the flesh, we are not waging war according to the flesh, for the weapons of our warfare are not of the flesh, but have divine power to destroy strongholds. We destroy arguments and every lofty opinion raised against the knowledge of God, and take every thought captive to obey Christ. There is power in the word of God. We must approach it humbly, or we could be burned as well. 1 Peter 3, 13 through 17. Now, who is there to harm you if you're zealous for what is good? But even if you should suffer for righteousness' sake, you will be blessed. Have no fear of them, nor be troubled. But in your hearts honor Christ the Lord as holy, always being prepared to make a defense to anyone who asks you a reason for the hope that's in you. Yet do it with gentleness and respect. And I hope I've had that attitude, because I have sought my heart and I said, don't get up here and be an arrogant jerk, because you have no right to do so, and you have no position in life to do so. And I hope that's communicated, and I hope it's going to be communicated on video, because I'm expecting that there's going to be some non-Creationists that watch this video. Do it with gentleness and respect, having a good conscience, so that when you're slandered, those who revile your good behavior in Christ may be put to shame. For it is better to suffer for doing good, if that should be God's will, than for doing evil. So, when Mr. Collins says, roughly, Adam and Eve do not fit the evidence, I say, Mr. Collins, what exactly did you expect to see? Have you ever taken the biblical data and said, what predictions would this make? Now, Francis Collins, if you somehow by some miracle are watching this video, I want to issue you a challenge, a direct challenge. A challenge to be like Joseph, like Daniel, like the Apostle Paul, great men of the Bible, and there are many examples, who are willing to risk everything for the name of Christ. God has elevated you, sir, to a very high position, a position that most people in this world will never attain. I certainly haven't. I'm a nobody in the scientific world. But I tell you what, I'm not going to stop doing this. My friends are going to continue to do this, and I don't think you're correct. And I don't know why you, as a believer in the blood of Christ, are attacking my position. I don't understand why it's such a threat to you. So I submit to you, and I challenge you. You have millions of dollars at your disposal. You know every single important person in the genetics world. You could tell a grad student, hey, grad student, why don't you go and do this project for me? You could get so much done that I can't get done on my piddly little laptop that locks up at 100,000 people. So I challenge you. I challenge you, sir, to explore the biblical alternatives, to see where they lead, and then take a stand, one way or the other, when you see the data and how they lie. God has put you in your position. He's put me in my position. He asks us to be fools for Christ. And that's what I believe we should all be trying to do. Thank you very much.